All right. So in last class, we started talking a little bit about principles of heredity. We introduced you the concepts of phenotype, genotype. We talked about dominant and recessive alleles. We talked about homozygous and heterozygous pairs of alleles. And we started talking a little bit about Gregor Mendel. Gregor Mendel, as I mentioned, is the individual who we credit with sort of elucidating a lot of the principles that we hold to be true right now regarding inheritance and the passing down of traits. And so Mendel and his pea plants are who we give credit to for a lot of the things that we know about being able to sort of predict the possible phenotypes of subsequent generations of breeding. And so Gregor Mendel did a number of breeding and crossings between these pea plants to examine different phenotypes. And, you know, he collected this data over years, and now we sort of have an idea of how we can predict what subsequent generations of organisms being reproduced what they will look like physically, or what their phenotypes can be. And so, you know, we refer to those, you know, the first generation as F1 generation, F2 generations. And he did a number of different crosses, and, you know, monohybrid crosses, dihybrid crosses. Monohybrid crosses where he's examining one factor, dihybrid crosses where he's examining two factors or traits. And like I said, we won't spend a whole lot of time on that in this class, but you'll definitely get a lot of practice with that when you get in your genetics course. And so these things we talked about on last class, we distinguished between genes and alleles. We know that genes are segments of DNA that code for a particular trait. And we know that genes exist in pairs that we call alleles, which means you can get different versions of that trait, right? Such as eye color. You can have blue eyes or brown eyes, and depending on which allele is the dominant allele, that's the one that you will observe in the phenotype. So hopefully you had a chance to go back and look over the things that we talked about on last class. You should definitely know the difference between the genotype, which is our set of genes, so G for genes, and your phenotype, your physical appearance, what you look like from a physical standpoint, your outward appearance is your phenotype. So those are some terms that we introduced on last class. And then we made this important concept point here that we inherit the genotype, not the phenotype. All right. We compared dominant and recessive alleles. We know that the dominant form of the allele is that which would be present in the phenotype, and the recessive form is masked or repressed in the presence of the dominant form. And so I think we left off here. Yeah, that the only time we will see the recessive allele present in the phenotype is in the absence of the dominant, which basically means that that's going to be a homozygous pair of recessive alleles for that trait. And so anyway, each organism, as I mentioned, there's going to be 2 copies of an allele for any trait. And these, depending on which allele that you inherit, or, you know, the dominant or recessive form, these separations are happening during meiosis, right? We talked about this being a sort of random assortment of genes or chromosomes during reproduction, and that process happens randomly, right? So this principle of segregation says basically that there's no sort of rhyme or rhythm to how these chromosomes and alleles are dispersed during crossing over. The concept of dominance tells us that when 2 different alleles are present in a genotype, right, that heterozygous pair of alleles, only the trait encoded by the dominant allele is observed in the phenotype. So this is now a principle that we hold to be true. We know this concept. Write this down. The concept of dominance tells us that whenever there are 2 different alleles present in a genotype for this given trait, only the trait that is encoded by the dominant allele is observed in the phenotype. You should be able to tell me that next week if I asked you about the concept of dominance on your exam. Okay. And so the experiments that Gregor Mendel did, as I mentioned before, he crossed up a number of different pea plants in a number of different setups in order to examine various characteristics. The first type of cross or breeding experiment that he set up was what we call a monohybrid cross, okay? And so just from a general standpoint, a monohybrid cross is a cross between two parents that differed in only one characteristic. Okay, so genetically, these parents are all the same except for this one factor. Okay, so for example, you know, we keep using this eye color, right? So you're crossing up two plants that have the same genetic makeup, except for, well, for the case of the plant, we'll say the shape of the seed, right? That's the example we've been using. And so everything else, all the genes are the same except for those that code for the shape of the seed, all right? And so the first generation of the cross is the P, right? That's that parental generation. And then the subsequent generations that will be produced, these offsprings, the result of the breeding or the crossing, are what we refer to as filial, F1 or F2, depending on how many generations are continued to be produced. And so he crossed up two homozygous pea plants for round seeds and a homozygous pea plant for wrinkled seeds. So there's a difference in only one characteristic. Okay, and so from a monohybrid cross, the offspring from the parent in the P1, in the P generation, which is parent, that first set of offsprings is what we would call F1. Okay, and so Mendel examined the F1 generations and found that they expressed only one of the phenotypes, right? All of the F1Cs were round, right? So when he did another round of crossing, right, he cultivated the F1 seeds, producing a second generation of plants, right? And both traits from the P generation appear in that F2 generation. So we had some that had round seeds and some that had wrinkled, right? And so after he continued to repeat that experiment over and over and over again, he realized that there was a pattern, there was a ratio for the presence of those two different phenotypes, right? So he realized that both of those traits were happening at a ratio of 3:1. All right. So we know that if we conduct monohybrid crosses, right, where these individuals are genetically the same except for one trait, right, that first F1 generation are all going to have the same phenotype. The F2 generation are going to present in a 3:1 ratio for the two different phenotypes that are possible, okay, in terms of dominant and recessive. So in this example here, we're looking at the flower plant color, the color of the plant, right? You have the purple parent and you have the white parent, which is the recessive. And so the capital P's tell us that purple is dominant. The little p's tells us that the white allele is recessive, okay? All of the individuals in that F1 generation are going to have the purple phenotype, okay? F2 generation, we're going to have a 3:1 ratio of purple to white. That's a standard that we now know to be true for monohybrid crosses where we're differing only in one trait or characteristic, all right? That's the monohybrid cross. This is just another example of a table where we set up a monohybrid cross. This Punnett square shows us that, again, we're going to end up with the phenotypic ratio of 3:1 after 2 rounds of breeding. All right, so we're crossing plants here that have both yellow and green, homozygous parents. One's yellow, one's green. So they're differing genetically in only one characteristic. All of the individuals in that F1 generation are going to have the yellow phenotype, okay? And the individuals or organisms produced in the F2 generation are going to have a 3:1 ratio of dominant to recessive phenotypes of yellow and green. And so I don't want you to, feel overwhelmed with this information, but just try and take some time to come back and read your text if you need to. And the exercise that you're going to work on today is a comprehension exercise for this as well. And so, things should click a little bit more if they aren't clicking right now. So 2 principles of inheritance that I want you guys to be familiar with, okay? The principle of segregation and the concept of dominance, all right? These principles of inheritance are those things that we now hold to be true as a result of those pea plant investigations that Gregor Mendel conducted. So the first one, the principle of segregation, says that 2 alleles separate when gametes are formed, and then so that process of crossing over takes place during meiosis. All right, we talked about that a little bit in our last lecture. All right, this random disbursement of our chromosomes and alleles happen during the process of meiosis, and those gametes are being formed. All right, gametogenesis. So 2 alleles can separate when those gametes are formed, because we know these gametes are only going to have 23 chromosomes. And so those pairs of alleles are separated during the production of these gametes. All right. All right. The concept of dominance. When 2 alleles are present in a genotype, only the trait encoded by the dominant allele is observed in the phenotype. We're clear about that now. That's a very clear concept in genetics and inheritance. Okay. The principle of independent assortment. When these gametes are formed, alleles of different genes separate independently from one another. So again, there is no connection, there's no rules, there's no way to guess or predict which alleles are going to be distributed in which way, right? There's no connection that if you get, allele A, that you'll end up with allele B for this other gene. All of this happens independently of each other. So the allele that one gamete receives for a gene does not influence the allele another gamete receives for that gene. All right, so it's all happening in a random fashion. Okay, so that's the concept of independent assortment. Okay. There's independent assortment. There's no association between, which gametes receive these genes or alleles for this gene and which ones the next gamete will receive. All right, so let's talk about these sex chromosomes for a little bit. Okay, so the sex chromosomes are going to be, the genes are on our sex chromosomes are the most important sex determinant in eukaryotic organisms, right? And so what we're saying here is that sex of an individual is generally determined by sex chromosomes, right? So we looked at that karyotype and we saw chromosome numbers 1 through 22. We said that those were for somatic cells. And then that last pair that had a letter designation of either XX or XY, those are our sex chromosomes, all right? There's one pair of sex chromosomes, right? These sex chromosomes are generally what's responsible for determining the sex of an individual, okay? If you're XX, you're female. If you're XY, you're typically going to have a male phenotype. Now, in some cases, right, we know that nothing's sort of absolute nowadays. In some cases, environmental factors can exert control over your sex, right? What you present physically with, right? But typically members of one sex have a pair of similar sex chromosomes. And so in normal cases, females are going to have a pair of sex chromosomes that are XX, and males are going to have a pair of sex chromosomes that are XY. Write that down. Okay. So again, females contain 2 X chromosomes. Male sex cells can have 1 X chromosome and 1 Y chromosome. What that tells us is that We see the X in both species, but the Y chromosome we only find in the male individuals. That means that the Y chromosome determines the maleness of the species. Okay, so males, all of the traits and features that are exclusive to the male are typically going to be coded for on the Y chromosomes. And so those are your sex organs, your penis, your scrotum, you know, things that are structures that are anatomically exclusive to the male are coded for on the Y chromosome. Okay, make a note of that. Those traits or characteristics are coded for on the Y chromosome, the maleness, all of the things that are exclusive to the male, right? So another point to make here is that we see the Y chromosome, X chromosome in both species, both male and female. And so that lets you know that the X chromosome is required for life. Write that down. Absence of the X chromosome is not a viable, will not produce a viable fetus. Okay. X chromosome contains genes that are required for both sexes. That's why we see the X chromosome in both pairs. Okay. A fetus with a Y chromosome and no X chromosome does not survive. Okay. So this would be a lethal reproduction of result. Okay. That fetus would not survive. On the other hand, a person with one X but no Y will have the overall appearance of a female, but with some defects. Okay. Because you're supposed to have two pairs of chromosomes there. So the absence of the X chromosome does not produce a viable individual. So if something happens during crossing over, right, during the formation of those gametes, and for whatever reason this fertilized egg did not get a X chromosome during independent assortment, that fetus will not develop. Okay. And we're making a note here that the Y chromosome contains 95% of the male specific traits. Okay. We're clear about that. So you should be able to have a general understanding of the sex chromosomes. Okay. What are the chromosomal pairs in the species for male and female? You know, what happens if this pairing doesn't occur the way that it should? Okay. Which of these chromosomes are required for life? X chromosome. All right, two more concepts, and then I think I'm going to be done. Incomplete dominance. Write this down. So when we are talking about patterns of inheritance and being able to sort of predict the phenotype in subsequent generations, things don't always happen the way that they should, right? We understand this concept of dominance. We understand the concept of dominant alleles versus recessive alleles. We can make these predictions, but things don't always happen 100% the way that we think that they should. Right? Incomplete dominance is an example of that. Dominance is not always complete. Now, theoretically speaking, we said that the dominant allele is the form of the trait that we're going to observe in the phenotype. However, that doesn't always happen 100% of the time, and a situation that we'll refer to as incomplete dominance, right? Incomplete dominance is when the phenotype that we see in that F1 generation is an intermediate between that of two parents. So for example, here I have some plants we're breeding. We've crossed a red parent and a white parent. All of the individuals in that F1 generation are going to sort of present as a mixture, a blend of the traits with mom and dad. So red and white in this case makes pink. Okay. This is the phenomenon known as incomplete dominance. Neither one of these traits in the parental generations were exerting complete dominance. Okay. So we ended up with sort of an intermediate. We know that white and red makes pink. Write that down. Incomplete dominance, when the phenotype of the F1 generation is an intermediate between that of two parents. Another situation where dominance isn't complete, it's called codominance. Okay, codominance is when the phenotype simultaneously expresses the phenotype of both parents. So again, with this plant here, we have white camellias, we have red camellias. The F1 generation of camellias are going to exhibit red and white phenotype, not a mixture, right? Not an intermediate where we saw in incomplete dominance. Incomplete dominance, you see red, you see white, and your F1 is going to sort of be a hybrid or a mixture, if you will, of red and white, and we get pink. In codominance, okay, both of these parental traits are behaving in a dominant fashion, and so we see manifestations of both of those traits in the F1 generation, both red and white, right? Not an intermediate, but we're seeing both of them being expressed simultaneously. There is a distinction. You should be able to distinguish between these two properties or characteristics. So there's another concept here. Pleiatrophy tells us that, and so generally speaking, we know that genes encode for a particular trait or that we see in the phenotype. Pleiatrophy happens when a single gene can have multiple effects on the phenotype. Okay, and so we see this in genetic diseases and disorders. Okay, where one gene can be responsible for a number of different symptoms or outcomes. Right, one pair of allele causes multiple symptoms. An example of that is cystic fibrosis. Okay. This abnormalities in this gene can present itself differently depending on the individual, where with cystic fibrosis, these individuals have abnormally thick mucus in different parts of their body, so respiratory congestion, digestive issues, reproductive system, things are just abnormal, and it manifests itself differently in different individuals, right? And so pleiatrophy says that a single gene can have multiple effects on the phenotype, not just one symptom. And this last concept that I want to tell you about is the norm of reaction. And so I started this conversation off earlier that, our genetic material, right, our DNA dictates the phenotype, like how we look and how we function, like what we appear, how we appear physically, but also from a physiological standpoint, a behavioral standpoint. Okay. And so for the most part, our genes dictate the things about us, our features, our hair color, and so forth. However, the environment cannot be negated. The environment also plays a role in shaping the phenotype of an organism. Okay. And so the norm of reaction is a situation where genes are interacting with the environment to ultimately influence what this phenotype looks like. And an example of this phenomenon is seen in hydrangeas. These are plants. And hydrangeas are very pretty plants, and you've probably seen them before. And you've probably seen them in different colors. What happens with this norm of reaction is that the genes are interacting with the environment, particularly the soil in this case, right? Plants grow in soil. You water them, they get sunlight, and things happen. So what happens with hydrangeas is that these plants are genetically the same, right? They're genetically identical. However, the flowers can range from a pink to a blue to a purple, and those colors that those plants bloom excuse me, are dependent on the contents of the soil before those flowers are planted. And so again, these plants are genetically all the same. They have the same genetic makeup. They have the same genotype. But depending on the pH of the soil that these plants are planted in, that these seeds are planted in, that's going to dictate whether or not those flowers are going to be pink or purple or blue. And so we will define the norm of reaction as the range of phenotypic possibilities, right? The range of phenotypic possibilities that can develop from a single genotype under different environmental conditions. Okay. And so in the case of these hydrangeas, I'm saying that we don't know if these plants are going to end up being purple, green, or pink, or whatever color. There's a range of possibilities from a phenotypic standpoint. These plants, though, are all genetically the same. These seeds all have the same genetic makeup. But depending on the pH of the soil, there is a range of phenotypic possibilities that these flowers can have. And so this range can be quite limited, or it can be very broad. All right. So for example, human height. This is a norm of reaction that can be very broad. You know, the human height can range everywhere from dwarfism all the way up to giant, right? So that's a norm of reaction where you have genes that are influencing the trait, but the environment can also play a role in the final outcome. And so this is just a picture of those hydrangeas that I just spoke of, all right, where we see the impact of the environment on phenotype. Okay, and so again, theoretically speaking, we say that our genotype determines our phenotype, but we're putting this asterisk here, right, to say that it's not always going to be 100% where our phenotype is exclusively determined by our genotype. We have to take into account the role that the environment has. And so this example here with the hydrangea plants shows the impact of the environment on the phenotype. Hydrangea flowers of the same genotype can range from blue to violet to pink, depending on the soil acidity. So again, these are two genetically identical plants, but if you plant them in soil that has different pH levels, then the colors of those plants are going to be different. All right, I'm going to stop right here. There are